Hi, everyone. Welcome to the PolyBio seminar series. My name is Amy Proal. I'm a microbiologist, and I'm also the research team coordinator for PolyBio. And we're a new nonprofit research organization working to innovate research on complex chronic inflammatory disease. So we started PolyBio because the era of an isolated scientist chipping away at the same hypothesis for a lifetime is over. Today's successful scientists must react, integrate, and build on cutting-edge discoveries across fields. But that isn't happening nearly enough. Most scientific knowledge is siloed in different labs without research teams that specialize in pulling key concepts together. And that segmentation creates continual blind spots in research, diagnostics, and even treatment. So we started PolyBio to change that. The way we work is our core team actively seeks out scientists with different backgrounds and different skill sets. Then we bring them together to conceptualize and conduct collaborative research on root cause drivers of chronic complex inflammatory disease. And our first round of research projects will focus on characterizing or defining a range of potential abnormalities in chronic conditions that start or are exacerbated by infection. These include myalgic encephalomyelitis, or MECFs, LongCovid or post-acute COVID syndrome, or PACS, and post-treatment or chronic Lyme disease. And these are conditions in which it's particularly important to unite the expertise of scientists at different labs and with different backgrounds, because as I hope you see in today's talks, the biological factors, the environmental insults, and the body systems implicated in MECFS and related chronic conditions, the infections, the microbiome, the immune system, host metabolism, the central nervous system, and the human genome are all themselves intricately connected. So if we really want to jumpstart research on these diagnoses, and at PolyBio that's our mission, we have no choice but to bring together virologists, pathologists, neuroscientists, molecular biologists, and other experts to make that happen. And that's the point of this seminar. Today, we hope to introduce you to some of these people, the scientists we have brought together so far at PolyBio. Some of the speakers are on our scientific advisory board, and others are working on collaborative projects or just good friends. And we're not presenting data yet from our specific PolyBio projects. We hope to do that at the next seminar. Rather, the point of today's seminar is to have talks that help you get to know our team so you can understand the truly innovative work they're already doing that can inform the study of MECFS and related chronic conditions. One moment. Okay, so as I said before, at PolyBio, we have a focus on studying chronic conditions that are initiated or exacerbated by infection. And unfortunately, the COVID-19 pandemic is providing a very visible example of this phenomenon in action. What's happening is about 10 to up to 30% of people who get COVID don't fully recover. Instead, they're developing a wide range of chronic symptoms that don't go away. And these patients are being given the diagnosis LongCovid or post-acute COVID-19 syndrome. And I'll call it LongCovid for the rest of this talk. Now, when this started happening, many people thought it was a new phenomenon. But for the last 8 years, I've been studying a condition called MECFS, and it's the exact same pattern. For decades, patients have been developing chronic symptoms after other infections. And many of those patients are given this diagnosis, MECFS. And in MECFS, the pathogens that initiate or exacerbate cases, in some cases, haven't been able to have been identified, but many cases do involve the herpes viruses or the enteroviruses, which are a family of polioviruses. And the central symptoms of MECFS are listed above. They interestingly overlap greatly with those that many long COVID patients are also reporting. And key symptoms are flu-like symptoms, malaise and sore throat, and these symptoms tend to relapse and remit. Also, a symptom called post-exertional malaise, in which patients cannot recover from even minor bouts of exercise. And finally, dysfunction of the autonomic nervous system, blood pressure issues, breathing rate issues, problems with fainting from moving just from a sitting to a standing position. And those are just a few of the core symptoms that these patients experience. So I want to clarify also that this trend, chronic symptoms after an acute infection, actually stems to most major pathogens. The literature is replete with examples of patients who develop a lifetime of chronic symptoms after infection with a tick-borne pathogens such as Borrelia, Bartonella, or Babesia, or other RNA viruses such as Ebola, Zika, dengue, or measles. And these conditions, these chronic symptoms after infection, they get different names, they get different labels. But the million-dollar question is why? What leads to chronic symptoms in a subset of patients? So I'll switch gears a little now. For the rest of the talk, I'm going to discuss trends that I think are important in the study of these post-viral or bacterial conditions by using long COVID as an example. And Mike Van Elziger, who's a neuroscientist who will speak after me, and I actually wrote a paper recently. It's not yet published, but was just accepted. So hopefully we'll be out there soon on different biological factors that can contribute to the development of persistent symptoms in long COVID patients. And so, I want to stress that all of the points that I'm going to bring up now also have direct relevance to MECFS or related, post-infectious diagnoses. So first, when it comes to long COVID development, we suspect that different biological factors may contribute to different long COVID cases, none of which have to be mutually exclusive. In other words, several problems may be happening in the same patient. And these are the top 6 factors that we discuss in the paper for chronic symptom development in these patients. One is organ injury. We know that the Sars Co2 virus drives a range of injury to many of the body's organs, and it's very possible that this injury does not heal over the long term and results in chronic symptoms. The second is that some patients may not fully clear the Sars Co2 virus from all body sites, and the virus may persist in reservoirs, especially in tissue or the central nervous system. Then, there's also the possibility that under conditions of immune dysregulation during acute COVID-19 disease, other persistent pathogens already harbored by the patient reactivate and themselves infect a new nerve or the central nervous system in a manner that can drive chronic symptoms. Also, since we're not sterile and we're full of organisms in microbiome and virome ecosystems in many of our body sites, it's possible that immune dysfunction and dysregulation, again, under conditions of acute COVID-19 disease, can allow a collective move towards imbalance in these microbiome and virome ecosystems that itself results in chronic symptom development. Finally, there may be autoimmune phenomenon at play, but I think it's very important, as I'll discuss more later, to consider the origin of the autoantibodies that may be identified in patients, because molecular mimicry between pathogen and host peptides contributes, in many recent studies, has been shown to be a real driver of autoantibody production in disease. And finally, and this was what Mike will talk about later, we're interested in how all these factors, these inflammatory or infectious insults, may be able to disrupt signaling of the brainstem or the vagus nerve in patients with these symptoms and diagnosis. So the first organ injury scenario I won't talk about today because that's being discussed by many other research teams. I want to mention a little bit more about point 2, the possibility that the Sars Co2 virus may not fully clear from all patients with a long COVID diagnosis and drive chronic symptoms. And this is a study that looked at got intestinal tissue biopsy samples from patients about 4 months after acute COVID disease. And they identified SARS-CoV-2 RNA and protein in 7 of the 14 biopsy samples. And there was a similar study done by another team that obtained nasal mucosa tissue samples, also from patients. It was about 6 months after acute COVID disease, and they had ongoing loss of smell. And that team identified high levels of SARS-CoV-2 RNA and protein in several of the tissue mucosa samples from the nose. And what's important to note about both these studies is that the patients who had the SARS-CoV-2 RNA or protein at time points that extended beyond acute disease did not test positive for SARS-CoV-2 or for COVID via conventional testing. They were all negative via normal nasal swab testing. So it took the analysis of these specific tissue biopsies to be able to find the viral RNA and protein at a later date. Now, these same kinds of studies have been conducted in patients with MECFS with very interesting results. This is a study done by a researcher named John Chia, who works out of California. And what he did is he obtained, similar to the other study I mentioned, intestinal tissue biopsy samples from patients, this time with an MECFS diagnosis. And of the 165 samples he tested, 82% of them stained positive for enterovirus VP1 protein. And many also had the RNA of the enterovirus in them. And that was compared to just about 20% of control samples that stained positive for enteroviral VP1 protein. And there are other studies on patients with MECFS that have, again, found enterovirus protein or RNA in other tissues from patients. So for example, there are multiple studies that have found enterovirus RNA or protein in skeletal muscle samples obtained from patients with an MECFS diagnosis. And there have been 2 autopsy studies in which patients' brains were shown to have enterovirus protein or RNA. So why don't we hear about this more? Sometimes I feel like the possibility of viral persistence is the least recognized. And I think one reason for that is that the fluids we commonly collect to study a disease process, the blood, urine, sometimes cerebrospinal fluid, they're not the fluids in which viruses and many persistent pathogens tend to stick around in. Viruses tend to clear from fluids over time where the immune response is the most robust, and they move into tissue or nerves. And in fact, several of the enteroviruses and all of the herpes viruses are neurotrophic, which means that they preferentially infect nerves, with the herpes virus active lifestyle requiring movement through nerves. And SARS-CoV-2 is similar. The virus is rarely found in the blood or even the cerebrospinal fluid, even in acute disease. So it also seems to prefer tissue, which is dense with ACE2 receptors, or a number of teams now have also found the SARS-CoV-2 virus in nerves, including even in the vagus nerve. So, these pathogens as well, they're often capable of getting into the central nervous system. They can sometimes breach the choroid plexus, or they can hijack immune cells in a Trojan horse-like fashion to gain access to the central nervous system of the brain, where there's even less of an immune response that they have to face. And the SARS-CoV-2 virus has been shown in several very good autopsy studies now. It's been found, its RNA and protein have been found in multiple brain regions in acute disease. And it does seem like it can also, the SARS-CoV-2 virus, enter the brain by infecting the olfactory epithelium of the nose that contains sensory neurons whose axons project into the olfactory bulb of the brain. So that may be another route by which SARS-CoV-2 can get into the central nervous system to drive symptoms that could potentially become chronic. So an overall takeaway here is, if you want to identify persistent pathogens in a patient with chronic symptoms, studies of fluid, blood, and urine are not your best bet. What you mostly want to do is very good autopsy work, and you also want to work hard to collect tissue samples from patients, either via biopsy or often surgery, where patients are undergoing surgery, provides a valuable opportunity to obtain tissue. And in fact, that is the basis of one of our main PolyBio projects. What we're going to do is we're collecting tissue biopsy samples that often contain nerves, or samples collected during surgery on patients with an MECFS or some related diagnoses, and we're going to analyze them for a range of pathogens and other issues. And we're working on that with David Pazaspino, who will speak later. So I also want to stress that the possibility that RNA viruses could persist in the central nervous system of a patient to drive chronic symptoms is not a new phenomenon. This is one of my favorite papers, and it's from the year 1986. It's called Persistence of RNA Viruses in the Central Nervous System. And the authors of the paper state, although it would seem difficult for RNA viruses to persist in the brain under conditions of normal immune defense mechanisms, representatives of at least 7 of the established families of RNA viruses have been shown capable of causing persistent infections under these conditions. So this is not a new thing. And in fact, I'm very excited for William Emer to speak later today about his lab's work on how amyloid plaque in the Alzheimer's brain may form in response to persistent pathogens in brain tissue in Alzheimer's. So another topic that can come up when teams find the RNA of a virus in a sample from a person with long-term symptoms is sometimes the findings are dismissed as not mattering much because it might be inert RNA. And to be fair, I've never really understood this argument, and a lot of people don't, because nearly every human cell type contains what are called RNase enzymes. They're enzymes that rapidly degrade inert RNA. So if the RNA in samples that we collect on patients is inert, the molecular mechanisms of this inert RNA persistence would really have to be much better delineated. And I think we can learn a valuable lesson on inert versus viable RNA from the post-Ebola syndrome community. So for a number of years now, we've known that, same pattern that I described before. Ebola patients, some of them develop chronic symptoms after Ebola infection. And if you test the blood, the fluids of these patients, you no longer find the virus or its RNA. However, multiple teams have looked in what are called anatomical sanctuaries in these patients, and they have found Ebola RNA. So for example, they found Ebola RNA in men's semen months or years after initial infection, and also in women's breast milk, and even in anatomical sanctuaries like eye tissue. So some of the first interpretation of these findings was that the RNA was inert, maybe it didn't matter. And then this happened just recently, just this January. This team published a paper, and they found that levels of neutralizing and total antibodies continue to fluctuate in the plasma of a high proportion of Ebola survivors. And the team contends that this periodic antibody resurgence might follow periods of low-grade Ebola virus replication in tissues that are shielded from a full-blown immune response. And so that really started to get people thinking that there might be viable virus in these patients. And then this happened just about a month or so ago. There was a new Ebola outbreak in Africa. And when the strain of the Ebola pathogen causing the outbreak was sequenced, several teams found that the strain from this outbreak barely differed from a strain seen 5 to 6 years ago on a different outbreak. And 3 independent research teams concluded that this new Ebola outbreak was likely sparked by a person infected over 5 years ago who had chronic viable Ebola virus in anatomical sanctuaries that was able to even start a new pandemic. So I think we need to be open-minded to the possibility that when we're studying long COVID or MECFS, and if we find viral RNA in tissue samples or in biopsy samples, that it may represent virus that is actually viable. So another trend that Mike and I talk about in our long COVID paper is that under conditions of COVID-19 disease, the immune system becomes dysregulated, and that may provide a very good opportunity for persistent pathogens already harbored by the patient to reactivate and drive new chronic symptoms. And all of us, the reason that this can happen is all of us harbor persistent viruses. And if you want a good example of that, this is a paper, the image on my slide is from a paper called A Tissue Level Atlas of the Healthy Human Virome. And what the team did is they collected over 50 tissue types from patients, healthy patients at autopsy, and they sequenced them for a range of viruses. And they found they detected 39 species at least one tissue type. And you can see on the left-hand column of the slide, some of the viruses that they found, again, in the tissue of these healthy subjects. So several of the herpes viruses, DNA viruses, but also RNA viruses, including, interestingly, several coronaviruses, not Sars CoV2, but other coronaviruses in this healthy tissue. And so what can happen is that a new infection is a blow to the host immune response. So for example, most major pathogens, and this includes SARS CoV2, they take down the immune system to better survive. So SARS CoV2 can disable T cell signaling, and the virus also expresses at least 10 proteins that block or mitigate interferon signaling. And interferons are cytokines whose activity inhibits viral activity and replication at many levels. So if you knock down the host interferon response, you knock down the body's first line of defense against most forms of viral infection. In fact, the activity of interferons plays an important role in keeping herpes viruses in latency. So overall, what can happen is that this immune dysregulation creates an excellent atmosphere for pathogens already harbored by the patient to reactivate and themselves potentially drive a range of chronic symptoms. They might infect a new nerve or get into the central nervous system themselves. And I want to clarify, this doesn't just have to be viral pathogens. Many people already harbor persistent fungi or persistent parasites or persistent bacteria, such as Borrelia burgdorferi. And those pathogens may also be able to reactivate under conditions of COVID-19 immune dysregulation. And I'm excited that Lena Pernis will talk later today about toxoplasma reactivation, a parasite, under conditions of severe acute COVID disease, which is a good example of this phenomena. So I want to stress another very important thing that most of these pathogens can do, a very important research consideration, which is that they hijack the metabolism of the cells they infect, which can drive a range of metabolic abnormalities in patients who harbor them. So this is another paper that Michael Van Azziger and I recently wrote and published in the journal Immunometabolism on that topic. And overall, what we describe is how most viruses and many bacteria are obligate intracellular pathogens. They rely entirely on the metabolic capacity of the host cells they infect to gain the raw materials for their replication and nutritional needs. So to obtain them, they hijack the mitochondria of the cells they infect in order to redirect glycolysis and TCA cycle intermediates towards the biosynthesis of lipid droplets, fatty acids, and nucleotides for their own replication and nutritional needs. And by definition, that changes the metabolic output of the infected cell. So this is a figure from our paper, and it shows how infection by different viruses alters different host cell metabolic pathways. And the great thing is that the cell and the mitochondria fight back against this hijacking. Mitochondria are now understood to function as part of the innate immune response. They have their own defenses that they use to combat these pathogens. And that's a key topic that both Clovis Palmer and Lena Pernis will be talking about later today. But overall, the takeaway is that when we study long COVID and MECFS, these conditions, we should be studying infection and host metabolism as interconnected topics. Now, another scenario for chronic symptom development in patients with long COVID is that we're not sterile. We all harbor trillions of organisms in our guts, in our mouths, in our lungs, in our bladders. In fact, most human body sites and even the blood do not appear to be sterile. So again, that immune dysregulation, the drops in T cell signaling or interferon signaling that occur during acute COVID-19 disease may allow organisms in these microbiome communities to increase their virulence. And that's because it's important to understand that most organisms in host microbiome or virome communities are what are called pathobinds. They can act as commensal organisms under conditions of balance or homeostasis, but they can change their gene expression to act as pathogens under conditions of immune dysregulation. So, for example, this team found that commensal E. coli, which is a very common member of microbiome communities, can evolve into virulent clones in less than 500 generations. So again, its immune dysregulation under conditions of acute COVID disease can promote a collective imbalance in the virulence of host microbiome communities in a manner that can result in a range of chronic symptoms. And it's also important to understand that if the microbiome of virome or various body sites becomes dysbiotic or imbalanced or pro-inflammatory, the barriers that contain those organisms, the gut lining, the epithelial barrier of the mouth can become more permeable. And that allows organisms to better leak into the blood or even into tissue, where then they can really drive a range of inflammatory symptoms. So for example, Marcelo Fiera will talk later today about the oral microbiome under conditions of COVID-19 disease. And bacteria or other organisms that accumulate in blood can also even drive hypercoagulation and amyloid formation, which is something that Riccia Pretorius will talk about later today. So the last topic I want to talk about for chronic symptom development in patients with a long COVID diagnosis is potential autoimmune phenomena. However, it's important to consider the origin of autoantibodies identified in both COVID and potentially long COVID patients, because many autoantibodies, what become called autoantibodies in the human body, are produced via molecular mimicry. And what I mean by that is most pathogen proteins can be very similar in size and shape to human proteins, human receptors, and human structures. So what that means is when the immune system tries to target a pathogen protein, it may also target a human protein or structure with a similar size and shape. It's collateral damage. And so because there are many pathogens that can persist in humans and definitely many pathobiont organisms that express a broad range of proteins in humans, the potential for molecular mimicry to contribute to autoantibody production is huge. So for example, quickly, this first study, the team found that B cells infected by Epstein-Barr virus had been shown capable of reacting with dozens of self-antigens, including albumin, thyroglobulin, and renin. Or it's well documented that Epstein-Barr protein, EBNA1, can cross-react with several of the lupus-associated autoantigens. And the second study is a good example of bacterial mimicry. The team tested for the presence of IgA or IgG autoantibodies directed against 14 human regulatory peptides, such as insulin, ghrelin, and leptin. And they found numerous cases of sequence homology between those peptides and the protein structures of over 30 bacterial species, including lactobacillus, E. coli, and listeria, which are common pathobinds in the human microbiome. So last, Mike will talk about this more, but I want to stress that we're very interested in how these different infectious and inflammatory insults may be able to impact brainstem signaling. And the reason we're interested in the brainstem in these patients is because nuclei in the dorsal brainstem control what's called the sickness behavior response. So the subjective feeling a person gets when they're sick that tells them that they're ill, that they should stay in bed is part of brainstem signaling. Also, brainstem signaling controls pain and nausea, and nuclei in the brainstem also control autonomic functions such as heart rate and breathing. And if those symptoms sound familiar to you, it's because many of them are the core symptoms in MECFS and long COVID patients. And I'm very excited that Roberto Slocco will later talk today about brainstem imaging so we can have more of a focus on that. And last, is it possible that the brainstem could be directly infected in patients with long COVID or even MECFS? I think it's an interesting consideration. This is one of the autopsy studies done on MECFS, an MECFS patient, and they did find enterovirus RNA in the brainstem of the patient. And this is an excellent autopsy study done by a team in Germany on patients with acute COVID. They did find, if you look at the green arrows on the left, those are viral protein positive cells in the medulla oblongata of the brainstem. And the picture on the right shows Sars CoV2 nucleoprotein in vagus nerve and cranial nerves originating from the lower brainstem. So I think that's something we could consider checking on in long COVID as well. And with that, I will hand this over to Mike. Thank you.